Welcome back to the Tuesday edition of Daytime. My name's Dan Mooney. There has been a research contest that has been put on by Diabetes Canada called End Diabetes Awards. And we have in our studio this afternoon a very special guest because this person has been awarded part of $9 million in diabetes research. She is Dr. Jennifer Bruin, an associate professor of biology and the Institute of Biochemistry at Carleton University. Dr. Bruin, thanks so much for coming in. Congratulations on winning this award. It's quite prestigious, isn't it?
Thank you. Yes. Yeah, it's a real honor to be selected with researchers from all across Canada. This competition is a really exciting one.
Now, your research is primarily focused in cancer.
Actually, this is just one very small and kind of new direction for my research program. So the broad theme that really touches on all of the projects is that we're interested in diabetes and why diabetes rates are increasing worldwide and here in Canada. And this particular research project is focusing on a unique group of patients who develop type 2 diabetes after having had cancer. But we're interested in, why diabetes rates are increasing across the board, not just in this unique subpopulation.
Is it because of the lifestyles that we all lead now in the 21st century?
So that certainly is one component. But what we're learning is that, there's more to it. It's complex and there's, a whole range of different factors that are contributing that we don't yet fully understand. So that's part of what my lab is trying to contribute to, is understanding, what factors in our environment, for example, are contributing to increasing risk of diabetes. You say that there's a genuine risk for cancer patients to contract type 2. Yeah.
Which is the most severe? 1, 2, 3, 4?
So in cancer survivors, and this is true for a number of different types of cancers, there's a well-documented increase in risk of type 2 diabetes after cancer.
Which is treatable.
Which is treatable, absolutely. But we're concerned about that increased risk because cancer survivors who go on to develop type 2 diabetes have worse health outcomes and they have higher rates of mortality. So if we can understand the connection between why somebody who has had cancer has an increased risk of developing type 2 diabetes, then hopefully we can mitigate that risk and design an intervention to prevent those patients from going on to develop diabetes. And so that's what our newly funded research project will be looking into.
Now, in the information that I've received before the show, you're working a lot with laboratory mice, and you're detecting things that you didn't see as recently as a year ago. How is that's such a rapid kind of information that you're able to work with?
So we've been working on this project now for a few years. It started with undergraduates in my lab as sort of a side project, something kind of an interesting question, which was, we're interested in the cancer drugs that patients have to take for their chemotherapy. Mm-hmm. And whether those cancer drugs, in this case we're looking at one called cisplatin, whether those drugs are having side effects and targeting the pancreas. So we started working on this a few years ago, and we started to generate some really interesting data to suggest that that cancer drug is potentially affecting insulin secretion. So the beta cells in the pancreas make insulin, and we need insulin to control blood sugar levels.
Is cisplatin something that's regularly prescribed for cancer patients?
Yeah, so it's commonly used to treat different types of cancer. And we know that there's side effects of these chemotherapeutics. That's unavoidable. But what we're trying to understand is whether these chemotherapeutics are potentially causing injury to the cells that make insulin. And we have some really interesting data that we've generated over the last few years suggesting in mouse beta cells that this drug disrupts insulin secretion and causes maybe death of a subset of beta cells, which is concerning because these cells don't have the capacity to regrow and regenerate like a lot of other tissues in our body do. So if there is that injury during chemotherapy, then that can put that individual at a higher risk of developing diabetes later.
Are there alternative prescriptions that people can get other than cisplatin to sort of eliminate this potential problem?
So that would be one solution, is finding a different type of chemotherapy that could be equally as effective. But the approach that we're taking is that, we want the best possible cancer treatment. So if it is cisplatin, and we know we can document how cisplatin is causing injury to beta cells, then we could potentially design an intervention that would protect those cells. So that's the other aspect of our funded research project, is we're trying to understand exactly how cisplatin is damaging beta cells, so we can design an intervention that could be maybe administered alongside cisplatin or shortly after to protect and preserve beta cells to reduce and mitigate risk of those patients going on to develop diabetes.
Having worked in a lab at one point in my life, I knew that there was no guarantee about any level of research that you were conducting.
Yeah.
How do you feel about the success of this particular experiment?
Yeah, so that's a really important point. So anytime we're working in a model system like a mouse, that's a basis to start our research questions. But in my lab, we're always trying to make sure that our work in a mouse model, for example, translates to humans. So one of the first steps of our Diabetes Canada funded project will be to make sure that what we're seeing with mouse beta cells translates into human beta cells. And so we have access to human beta cells through the University of Alberta, through a donor program. And so that's one of the next steps for us, is to make sure that what we're seeing translates into human beta cells.
Wonderful. Dr. Jennifer Bruin, Associate Professor, Carleton University, congratulations on your award, and thanks for coming in today.
Thank you.
More Daytime coming up.